Hello, this presentation is titled Glycolysis, Gluconeogenesis, and the Role of Aldolase Gene Expression. In this talk today, I will be describing two different metabolic pathways, the first being glycolysis and the second being gluconeogenesis. Following that, I will talk about the different types of aldolase enzymes that exist and based on their location throughout the body. And then I will go into some of the different transcription factors that control aldolase B, which happens to be the aldolase form in the liver. This presentation is for Grand Canyon University by myself, Catherine Conan. This is for Chem 530, Concepts in Biochemistry. And the professor for this course is Dr. Jing Li. First thing that I'm going to go through is just a lot of information about glycolysis. So glycolysis is the main metabolic energy pathway throughout the body. And it is the way that our body breaks down glucose to produce energy. It is what's the first step in cellular respiration. Following glucose, eventually the products of glucose will then enter the tricarboxylic acid cycle and eventually the electron transport chain, which will produce a lot of energy for each cell. The glycolysis cycle deals with the oxidation of glucose. Glucose is a 6-carbon hexose sugar. It is a monosaccharide, as you can see. It is cyclic as well. There is an aldehyde group on carbon 1, and carbons 2 through 6 contain hydroxyl groups, which are the OH groups that you can see. Glycolysis breaks down glucose, which is the essential fuel for all animals, plants, and microbes. It is found in all different types of cells throughout the body, and that is the way that the cells make energy to stay alive and to be able to divide and grow. Now here is the actual 10 steps of glycolysis, starting with a single glucose molecule. There is a commitment step that occurs first, in which ATP is actually burned, and that is the commitment step with hexokinase. This is an irreversible reaction. Once glucose enters the cycle, it is then converted with ATP to glucose 6-phosphate. So if you notice, there is a phosphate group that has been attached to the molecule itself. Continuing on, glucose 6-phosphate turns into fructose 6-phosphate. Another ATP is burned to take us to the fructose 1,6-bisphosphate. Now at this point, this is where the cleavage occurs, where the 6-carbon chain breaks down into 2 different 3-carbon chains. And you can see with the double arrows here, we end up with a glyceraldehyde 3-phosphate and a dihydroxyacetone phosphate. Now the dihydroxyacetone phosphate can then be converted through an additional enzyme into a glyceraldehyde 3-phosphate. So eventually, into the second half of the cycle itself, we end up with 2 glyceraldehyde 3-phosphates going in. And now we're going to actually start to get a little bit of energy from the cycle. The first half is a commitment phase, or what is known as the investment phase. 1 glucose goes in, and we use 2 ATP to get the cycle started. And then the second half is known as the payoff phase, and that is what we're going to go into next. In this case, we have to double all of the rewards that we get for the second half of the phase because 2 of those glyceraldehyde 3-phosphates are entering the second half. So everything has to be doubled. But in this step here, glyceraldehyde phosphate dehydrogenase actually reduces an NAD+, and so we get some NADH out of the reaction, which is, again, a carrier of our electrons. Continuing forward, eventually we break that down into 1,3-bisphosphate glycerate, get an additional ATP, 3-phosphoglycerate, 2-phosphoglycerate, and then eventually we get an additional ATP on our way to make pyruvate. Pyruvate is the final molecule that is produced by glycolysis. So, just to recap, the cost for glycolysis to break down a 6-carbon sugar, like glucose, is we're going to use 2 ATPs. Once we have cleaved that 6-carbon that 6-carbon glucose into 2 3-carbon chains, we get some NADH coming out. We get 2 of those, and then we get 4 ATP. Now, this cycle doesn't produce a ton of energy. As you can see, we're only getting 4 ATP out of it, and it cost us 2 to start. All of that energy really comes later in the electron transport chain. But that is for a later presentation. Now, we just went through glycolysis, and there are actually 3 steps of glycolysis that are irreversible. And these are commitment steps. Once we bypass this, we can't go back. The first one is the hexokinase conversion of glucose to glucose 6-phosphate. Then there's also a step where fructose 6-phosphate, with the help of phosphofructokinase 1, produces fructose 1,6-bisphosphate. That's right before the cleavage step. That's also an irreversible step. Last but not least, there is an irreversible step with pyruvate kinase, the last step of the cycle itself, where pyruvate is produced. Now, there are times during an organism's existence where maybe they become low in glucose. Maybe they haven't been eating, maybe they have been exercising for an extremely long period of time. Maybe there has been a long-term sleep going on, but there may be situations where food is scarce, and so we have a pathway to counteract that lack of glucose that may be not happening in the bloodstream due to circumstances. And so because of that, our bodies have evolved to run a cycle called gluconeogenesis. Now, gluconeogenesis is not the complete reversal of glycolysis, because as I said, there are irreversible steps. But in gluconeogenesis, there are actually some enzymes that bypass those irreversible steps and allow us to make our way from 2 pyruvates back to the original glucose molecule. This can be used then to increase the amount of glucose in the bloodstream and keep certain organs that rely on glucose specifically, like red blood cells, different cardiac cells in the heart, different tissues like that need glucose all the time in parts of our brain, etc. And so we need to be able to bypass those irreversible steps. So some of the enzymes that can do that is a PEP carboxykinase, pyruvate carboxylase enzyme. This fructose 1,6-bisphosphate can work as a bypass step, and the glucose 6-phosphatase can work to counteract the irreversible step of the hexokinase. So, gluconeogenesis. I mentioned some of the tissues that rely specifically on glucose earlier. Kidneys, kidney cells, testes, different red blood cells, parts of the eye, and the brain. When the levels get low, prolonged fasting, overnight sleep, vigorous exercise depletes our glycogen stores, which our body needs around 190 grams of glycogen per day. There is about 20 grams stored in body tissues, but whenever that level of glucose gets low, this pathway can kick in. Now, there are several different precursors that can enter gluconeogenesis. Some of them come from many different parts in the body, and I'll show you that on the next slide. But glycerol, pyruvate, and lactate can obviously come in this direction and start eventually being converted to malate and going up to glucose from there. Propionate, as well as some amino acids, can be broken down, and especially in starvation situations, your body uses fat first. Well, first it uses free glucose, then it starts breaking down your glycogen. Eventually we get to fat stores, breaking down triglycerides, and then thirdly we start breaking down muscle, and then lastly, potentially organs, and you're in a world of hurt at that point. Now, gluconeogenesis takes place primarily in the liver and in the renal cortex. not all cells are capable of doing gluconeogenesis, okay? So, as you can see, 2-pyruvate go in, and is converted to malate, and then we can go through the gluconeogenesis pathway from there. Now, as I said earlier, some of these glucose, eventually what becomes glucose sources, come from different parts of the body. For example, there is glycogen stored in the muscle tissue that can be broken down. Glutamine is also found in muscle tissue that can eventually enter the cycle. It is freed up. There is also aniline that is present in gut tissue, etc. Proteins can be broken down into pyruvate. Glycogen can be broken down into lactate. All of these things can then enter the liver and enter gluconeogenesis through different chemical reactions. We also talked about adipose tissue, which is your fat tissue. That can be broken down into glycerol as well and enter gluconeogenesis, eventually to be broken down into glucose. That is why fasting a lot of times ends up breaking down fat, if that's something that dieters try to do. But eventually, these are all ways that we can follow different chemical reactions to get free glucose. So this also is an additional pathway slide showing that adipose tissue can break down glycerol in the liver. Through glycerol kinase, we can get glycerol 3-phosphate, and eventually that can be turned into DHAP, which can then enter at this step and enter the pathway going up to create glucose. Fructose, again, can also be turned into DHAP and enter the pathway at that point. Odd chain fatty acids, propionate, that can actually be turned into succinyl-CoA to malate to oxaloacetate and enter. Lactate from the Cori cycle can come in through lactate dehydrogenase into pyruvate and enter gluconeogenesis. And glycogen and galactose can enter and just has a very short pathway to get to glucose. So lots of different ways that we can provide that glucose for the body to be able to stay alive. Now, previously I also mentioned that certain amino acids can enter gluconeogenesis and produce glucose for us. Now, there are a couple different amino acids that cannot actually be used for gluconeogenesis, and that is lysine and leucine. Based on their chemical structure, they cannot come in and enter the cycle. But all of these other ones are available. Histidine, glutamine, valine, tyrosine, aspartate, etc. can all enter during parts of the citric acid cycle, which is the TCA cycle, Krebs cycle, etc., and then can eventually be converted into glucose. So these are called glucogenic amino acids, and the majority of them are able to produce glucose to keep different cells in the brain going in terms of reduced glucose.